Hello everyone, welcome back to Studious Students. In today's video, we'll learn about Entamoeba histolytica, an important protozoan parasite that causes amoebic dysentery and can even lead to liver abscess. We'll cover its morphology, life cycle, pathogenesis, diagnosis, and treatment in a simple and exam-oriented manner. Now that we know what organism we are discussing, let's begin with a brief introduction. Entamoeba histolytica is a pathogenic amoeba responsible for amoebiasis, commonly presenting as amoebic dysentery. It is found worldwide, but it is especially common in tropical and subtropical regions where sanitation may be inadequate. Humans are the primary host of this parasite. To understand how this parasite causes disease, let's first look at its different forms. Entamoeba histolytica exists in three forms. First is the trophozoite, which is the active feeding stage. It possesses pseudopodia for movement and may contain ingested RBCs. Second is the precystic stage, an intermediate form. Third is the cyst, which is the infective and environmentally resistant stage. Among these forms, the cyst is especially important because it initiates infection. Let's see how. Humans acquire infection by ingesting the mature quadrinucleate cyst through contaminated food or water. This mature cyst is the infective form of Entamoeba histolytica. Transmission occurs through the fecal-oral route. Once swallowed, the cyst begins its journey through the GIT tract. The cyst survives passes through the stomach because it is resistant to gastric acid. It then reaches the small intestine unharmed. This ability allows the parasite to successfully establish infection. After reaching the small intestine, an important event called excistation takes place. In the small intestine, the cyst undergoes excistation. A single mature cyst releases four trophozoites. These trophozoites are the active form that continue the life cycle. The newly released trophozoites now migrate towards their preferred site in the large intestine. The trophozoites move to the ileocecal region, especially in the cecum. Here they multiply and colonize the intestinal mucosa. This colonization is the starting point for disease development. However, not all infections follow the same course. Let's examine the possible outcomes. Entamoeba histolytica infection can have three outcomes. First, asymptomatic infection where no tissue damage occurs and cysts are passed in stool. Second, amoebic dysentery, where trophozoites invade the intestinal mucosa, causing ulceration. Third, invasive disease, where parasites enter the portal circulation and spread to the liver, causing amoebic liver abscess. The parasite achieved this invasion using several virulence factors. The first virulence factor is the amoebic lectin antigen, which helps trophozoites attach to intestinal cells. The second is amoeba 4, a molecule that forms pores in host cell membranes. The third includes cystine protease and hydrolytic enzymes, which aid in tissue destruction and invasion. Using these virulence factors, the parasite damages the intestinal wall and causes characteristic lesions. The trophozoites first colonize the large intestine. They attach to the mucosa through lectin-mediated adhesion. This leads to tissue destruction and the formation of the classic flask-shaped ulcer, a hallmark of amoebiasis. Some trophozoites then enter the portal circulation and spread to extra-intestinal sites, particularly the liver. These pathological changes give rise to several clinical manifestations and complications. Patients with amoebic dysentery typically pass bloody stools mixed with mucus and pus cells. The stools often have an offensive odor. This condition results from ulceration of the intestinal mucosa. Rare complications include fulminant amoebic colitis, amoebic appendicitis, and intestinal perforation. Although uncommon, these conditions can be life-threatening. The disease may also produce other severe extra-intestinal manifestations. Additional complications include toxic megacolon and amoebiasis cutis. The most important extraintestinal complication, however, remains the amoebic liver abscess. Now let's see how this infection is diagnosed in the lab. The simplest diagnostic method is stool microscopy. Wet mount examination can demonstrate both cysts and trophozoites. Cyst may show 1 to 4 nuclei, while trophozoites may display pseudopodia with ingested RBCs. If tissue samples are available, histological examination can provide additional evidence. On PAS or H&E staining, trophozoites can be identified within tissue. The presence of ingested RBCs inside trophozoites is highly suggestive of Entamoeba histolytica infection, as you can see in this image. Besides microscopy, the organism can also be grown and identified using specialized techniques. Entamoeba histolytica can be cultured using arsenic culture media. Although not routinely performed, culture can aid in research and specialized diagnosis. Modern lab methods further improve diagnostic accuracy. For serological diagnosis, we use ELISA tests to detect antigen and antibodies of amoeba. For molecular tests, we use nested multiplex PCR and real-time PCR, which targets the 18S rRNA. We also use biofilm assay. Asymptomatic carriers are treated using luminal agents, which includes idoquinol and paromomycin. For amoebic dysentery, we use metronidazole and the same luminal agents mentioned before. This is the pro table which summarizes all the high yield points covered in this video. You can take a screenshot if you want. Thank you for watching. If you found this video helpful, don't forget to like, share and subscribe to Studio Students. See you in the next video.